AS level biology 9700. Paper 1, variant 1, October, November 2024, question number 1 to question number 20. And question number 1 is, the diagram shows a transport section to a blood capillary. They have shown a blood capillary with the wall of endothelium and lumen has been shown over here, and the actual diameter is 7 micrometer. So what is the magnification of the drawing? So we have to find out the magnification, and you know the formula of magnification is equal to magnification. Some of my students told me that my handwriting is not clear when I write during my videos. Please try to understand that I always write on the screen of the laptop. That is the reason it is not clear. But try to listen my videos. You can see everything is explained into it, and it will be helpful for you to understand the answers. Thank you. Now you can see magnification is equal to diameter of drawing divided by actual diameter. So magnification M is equal to, they have given us the actual diameter, which is 7 micrometer. So we have to find out the diameter of the drawing, and the diameter of the drawing can be measured by measuring this line from here to here, and I have measured it. It is 35 millimeter. So we have to change it into micrometer. So 35 will be multiplied with 1000. So it will be equal to 35,000. So the answer will be 5,000. So D is the correct answer. So magnification is equal to 5,000. So D is its correct answer. Now, question number 2. And question number 2 is, which statement explain why lymphocytes with no nuclei die? So the cells do not have centrioles and cannot divide. Lymphocytes with no nuclei. So no nuclei, it means cytoplasm is there. So centrioles are present in the cytoplasm. So the first statement is not correct. The cells do not have mitochondria. Again, the mitochondria are always present in the cytoplasm. The cells do not have messenger RNA and cannot transcribe DNA. So messenger RNA is formed during the transcription and from the template DNA strand. So again, this is not the correct answer. They do not have ribosomes and cannot synthesize protein. They do not have ribosomes. Yes, the ribosomes are formed in the nucleolus inside the nucleus. So that is the reason, if there is no nuclei, that there will be no ribosomes, and if ribosomes are not there, then the protein synthesis will not take place. So D is the correct option. Third is the density gradient centrifugation is used to separate cell structures by their relative density. Larger cell structures have greater density and sink further down the centrifuge tube. What is the correct order of the cell structures starting from the top of the centrifuge tube? So we have to start from the you can say the lightest So what is the cell structure? lighter cell structure. So in this case, first of all, I would like to explain that what is the density gradient centrifugation. So this is the method by which the cell structure separation can be done. So when it is started, when this process is started and centrifugation tube into the centrifugation tube, or other words we can say the when the tube is spun, cell structures will migrate and settle at the point in the gradient where their density matches the surrounding medium. So in this way, the order of cell structures which will based on this density will be, first of all, the ribosomes. They will be smallest, being the smallest and least dense, ribosomes will settle at the top of the gradient. So it means A and B are not correct options. So C or D can be the correct options. So we have to find out the correct one. So after ribosomes, there are the mitochondria. And mitochondria, they produce energy. So they are denser than the ribosomes. So mitochondria are denser, so chloroplasts are not. And then chloroplasts are denser than mitochondria. The process of photosynthesis takes place in the chloroplast. And nuclei. Nuclei, they have chromosomes inside. So they are more denser than all other cell structures. So according to this arrangement, density of their density, the D is the correct option. A, B, C are not correct options. So D is the correct option. Now we will move towards the question number 4. And question number 4 is, which cell structures contain nucleic acids? Nucleic acids. So you can see that the first of all, the nucleic acid is present into the ribosomes. DNA is present, or you can say the chloroplast. They also have nucleic acid inside. Whereas the lysosomes have enzymes and Golgi bodies are used to modify, modify the proteins. So ribosomes are helpful in the uh, protein synthesis. So chloroplasts are also there, which contain the nucleic acid. So answer is C, 1 and 4. Golgi bodies, they modify the proteins. And lysosomes, they produce the enzymes. So C is the correct option. A, B, and D are not the correct options. Question number 5. And question number 5 is, the statements describe the processes that takes place in a secretory vesicle. Cell modification of the protein occurs in the Golgi body. Messenger RNA leaves the nucleus. Ribosomes bind to the messenger RNA during translation. Transcription of a specific DNA sequence occurs. Vesicles fuse with the cell surface membrane. Vesicles transport the proteins to the Golgi body. Statement 4 is the first process and statement 5 is the last process. So what is the correct sequence of the middle 4 processes? They have given 6 statements, and out of 6 statements, 2 statements, one of them is the first statement and the fifth statement is the last. So it means we have to arrange the middle 4 statements. So actually they have shown in this processes the protein synthesis and the so in this first statement, which is transcription of a specific DNA sequence occur. So process of transcription takes place in which a messenger RNA is copied from one DNA strand, or which is called as template strand. So second statement, messenger RNA leaves the nucleus. When transcription occurs, the messenger RNA leaves the nucleus through the nuclear pore. So second statement is this one, messenger RNA leaves the nucleus. And then the statement, ribosomes bind to messenger RNA during translation. So the second stage of protein synthesis takes place when messenger RNA leaves the nucleus. The messenger RNA binds itself on the ribosomes for the second stage of protein synthesis, which is translation. So this is the third statement. So then after this, the first statement, uh, sixth statement, vesicles transport the proteins to the Golgi body. So, and then modification, uh, yes, the vesicle transport the proteins to the Golgi bodies. So when the protein is synthesized, then vesicles transport that protein to the Golgi bodies. So this is the fourth statement. And then fifth statement is the which is left behind is the modification of the protein occurs in the Golgi body. It is the function of the Golgi bodies to modify the protein which is present in them. So this is the fifth statement. So this is one statement and this is the sixth statement. So according to this, A is not the correct option. C is also not correct option. D is also not, only B is the correct option. So answer of fifth question is B. Now question number 6 is, how many types of structures with a double membrane that are found in animal cells are also found in the plant cells? In animal cells, the nucleus, and then double membrane mitochondria, chloroplast. So chloroplast is not present in animal cell. So only nucleus and mitochondria are there, which are present in both animal and plant cells. So the answer for 6 is D. We have to, we have to answer which are both present in animal cell and in plant cell. So B is its correct answer. So a naturally occurring polysaccharide synthesized in a plant is a branched chain of alpha glucose. So the straight parts of the molecules are linked by alpha 1,6 glycosidic bonds with only a small number of branches, which are linked by either alpha 1,3 glycosidic bond or an alpha 1,4 glycosidic bond. So the which polysaccharide has a structure more similar to that described. So glycosidic bonds are formed by the condensation reaction. So when a water molecule is removed, then condensation reactions takes place, and as a result of condensation reaction, the glycosidic bonds are formed. So during the formation of these bonds, you can say, so different types. First is amylopectin, amylose, and cellulose, and glycogen. Amylose, amylose is a straight chain. It is not branched. So they have asked about the branched chain. And cellulose is also not, it is formed of beta glucose. So they have branched chain of alpha glucose. So B and C are not correct answers. Amylopectin is a branch structure, which can be the correct option. Glycogen is also branch structure, but the glycogen is more complicated branch than the amylopectin. So A is the correct option. D is not the correct option. So amylopectin, amylose, They are the two different types of structure which are part of the starch molecule. So amylopectin is the branch structure which have 1,6 glycosidic bond and as well alpha 1,4 glycosidic bonds. Glycogen, it is similar to amylopectin, little bit similar to, you can say, but it is more branched as comparison to amylopectin. So A is its correct option. Now, question number 8. And here you can see the question number 8. The diagram shows a triglyceride. Here you can see a triglyceride in which three uh, chains are there. You can see there is no double bond in this chain. One single bond is there and one double bond is there. And in the third chain, there are three double bonds. So an enzyme was used to digest these triglycerides in glycerol and fatty acids. You know, the triglycerides are formed with one glycerol and three fatty acid molecules. So we have to discuss these A, B, C, and D. First of all, you can see in the chain one, there was no double bond. So number of carbon double bonds and number on one side they have given the number of carbon double bonds, on other side the number of carbon atoms. So the number of carbon atoms and the number of double bonds. So we have to find out. So in the chain one there was no double bond. So A is not the correct option. But here in this case you can see the B, there is no double bond in the chain one. There is one double bond in the chain 2 and there is three double bonds in the chain 3. So B can be the correct option. Let's see the C and D. We are moving towards the C and D. And here you can see in C, you can see the one double bond is there. So C is not correct. No C. It means there is, you can go back to the, there is no double bond in chain 1. One in the chain 2, there is one double bond. In the chain 3, there is three double bonds. So according to this information, our correct option is B. So B is the correct option because according to this, zero double bond in chain 1 and one double bond in chain 2 and three double bonds in chain 3. So A, C, and D are incorrect options. Only B is the correct option. You have to understand this graph. Very easy. This is very easy. You just focus on the graph. Then you can find its answer. Well, the next question is, the diagram shows how the alternating nature of beta glucose monomers along a chain allow hydrogen bonds to form between consecutive monomers. Hydrogen bonds are sometimes, they are the weak bonds, but they are strong when they are present between some of the molecules, and they give hydrogen bonds in cellulose affect the tensile strength, the ability to withstand pulling forces without breaking. So how do the hydrogen bonds shown in the diagram help cellulose function as a suitable material for a cell wall? Okay, so you can see these are the hydrogen bonds. So the hydrogen bonds add additional tensile strength along individual cellulose molecules within the cell wall. Well, so the hydrogen bonds, again, understand this hydrogen in cellulose affect the tensile strength to withstand pulling forces without break. Okay, so hydrogen bonds add additional tensile strength along individual cellulose molecules within the cell wall. It can be a correct option because this is true. That is, the hydrogen bonds provide the additional tensile strength along individual cellulose molecule. And then the hydrogen bonds create stronger cross links between adjacent cellulose molecules. The adjacent cellulose molecules make the glycosidic bonds. So this is not the correct option. So the stronger hydrogen bonds formed between adjacent cellulose, again, they are not formed between two molecules. They are glycosidic formation of glycosidic bonds. So cellulose molecules within a cellulose microfibril have a stronger link. Now they are talking about the microfibril have a stronger link between them. So increasing the tensile strength. They are not talking about the hydrogen bond. So A is the correct option, which is correct. That is, the hydrogen bond add additional tensile strength along individual cellulose molecules within the cell wall. So this is the correct option. Now we are moving towards the next question, and next question is 10, and two solutions 1 and 2 each contain a mix of two different biological molecules. One solution contains starch and sucrose, and the other contains glucose and protein. And students know, for starch, there is iodine test, and for sucrose is non-reducing. Glucose is dienetic, and for protein, there is burette test. So these are the four tests. Okay, two solutions were tested with a variety of one group and the second group. Okay, so the two solutions were tested with a variety of reagents to identify the presence of the biological molecules in the solution. The table shows the results recorded for the various tests. Which row identifies the two solutions? Add iodine solution. Iodine is used to test the starch. So you can say that starch. So starch will give the positive test with a positive result with iodine. So, and whereas Benedict test and Buer test will give the negative results. So this is the this and the boil with Benedict solution, and here in this case the Benedict solution or the glucose will be positive test for Benedict solution. So glucose will give the positive in the group 1 iodine test, non-reducing sugar, sucrose, starch, they will give the negative. So this is the correct. While with Benedict solution after acid hydrolysis, non-reducing sugar after acid hydrolysis, the test 1 will give the positive results, and solution 2 will also give the positive result, whereas glucose is also there. Now, because they have to do the Benedict test as well. Add burette solution. Only the protein will give the positive result. So according to this, all these are the good. So C is the correct option. A, B, and D are not the correct option. Only C is the correct option. Now question number. Now you can see the diagram shows three pairs of amino acids three pairs of amino acids within a protein. The pairs are labeled X, Y, and Z. So X, Y, and Z, which row shows the correct type of interaction that would occur between the two amino acids in each pair. In amino acid, each pair. First of all, the X, you can see the bond is formed between oxygen and hydrogen is a hydrogen bond. So the second Y, you can see both amino acids in non-polar, they have non-polar side chains, CH3, CH2. So the bond which will be formed between these two amino acids will be hydrophobic interactions. So there will be hydrophobic interaction. Water haters, hydrophobic water haters. Now in the third, you can see the positive and negative. So it means positively charged side chain NH3, while the other has negatively side chain. So it means there is the formation between them is ionic bond. So according to this, the answer is hydrogen bond, hydrophobic interactions, and ionic bond. So B is the correct option. A, C, and D is not the correct option. Now we are moving towards the next question, number 12. And question number 12 is, a student carried out investigation at pH 1 to 8 to look at the effect of pH on enzyme catalyzed reaction. The optimum condition for this enzyme is the acidic environment of the stomach at pH 1 to. The remaining substrate concentrations were measured after 5 minutes at each different pH. After 5 minutes at each different pH. So here you can see the results. Which graph shows the effect of increasing pH on substrate concentration? Substrate concentration remaining after 5 minutes. So pH, stomach pH is between 1 and 2. It means the proteins are digested best into the stomach when the pH is highly acidic, or 2, 1 or 2. As the pH will decrease, we can say the pH will increase, the rate of digestion will decrease. So here substrate concentration remaining after 5 minutes, and on one side, and pH has been shown, which is between 1 and 8. So according to this, 1 and 2 is required. So you can see the substrate concentration remaining behind is very low, very less concentration is left behind. It means most of the concentration, substrate concentration, had been digested because it was appropriate or accurate pH for the digestion of the substrate molecule. And as you can see, the substrate concentration uh, is increasing as the pH is increasing. It means the substrate concentration is increasing as pH is increasing. So it means digestion is not taking place as the pH is not acidic. It is going towards the alkaline nature. So the digestion is not taking place because the enzymes are specific in nature. pH is specific for the digestion of the enzyme. So here proteins or the substrate molecules which has been given over here only digest at low pH. As the pH is increasing, the rate of digestion decreasing. So substrate concentration remain behind too much, you can say. And the other, first it is correct, but it is increasing and then decreasing. So B is not the correct option. We are moving towards the C option. And C option Again, in C option, first is not correct. You can see it is highest and then it is. So here in this case, you can see A is the correct option because the pH, required pH for the digestion of the substrate is 1 or 2. So at 1 or 2, digestion rate is highest, then substrate concentration is increasing as pH is increasing. So A is the correct option. Only A is the correct option. C, B, and D are not the correct options. So moving towards the next question. Question number 13. Which statement is correct for a non-competitive inhibitor? Okay. Well, students, you know, non-competitive inhibitor. Non-competitive inhibitor is that which do not attach itself on the active site. It attaches anywhere on the enzyme. So the inhibitor binds to the active site of the enzyme and decreases V maximum. Uh, it do not attach itself on the active site. So A is not the correct option. Inhibitor binds away from the active site and increases the Michael-Menten constant. No, it does not happen. Michael-Menten constant is not increased in this case because when it attaches itself anywhere on the from the active site, it is does not attach itself on the active site. It attaches anywhere on the enzyme. So in that case, it do not increases the Michael-Menten because it reshape the active site at that time. Due to de-shape of the active site, the process is stopped. The inhibitor uh, decreases the maximum, but the Michael-Menten constant does not change. It can be a correct option because uh, the reaction rate will be decreased because there will be, you can say, the reaction rate will be decreased. Why decrease? Because it will change its shape. So enzymes activity will be reduced. But here you can see uh, its Michael-Menten constant will reflect the same. It will not change. So C can be the correct option and D is the inhibitor does not change V maximum but increases the Michael-Menten. No, it will not. It will not because the Michael-Menten constant which reflects the substrate concentration at half of V maximum remains unchanged. So answer is the C because the V maximum will decrease but Michael-Menten constant will not change. So C is the correct option on. Pyrophosphatase enzymes catalyze a hydrolysis reaction. Pyrophosphatase ions, pyrophosphatase enzyme, phosphate ions product. So in experiment on a scientist studied the rate of this reaction using a calorimeter. The absorbance of the solution was measured at regular intervals until all of the pyrophosphate ions had been converted into phosphate ions. In experiment 2, the scientists repeated the procedure with a higher concentration of pyrophosphatase. They have increased pyrophosphatase. They have increased the enzyme concentration. All other variables were standard. All they were same, but substrate concentration was same, but they had increased the concentration of the enzyme. By increasing the concentration of the enzyme, students, you understand that the active sites will increase, whereas the concentration of the substrate will remain the same. If we increase the, as for example, you can say if there is an increase in the active site. So more substrate molecules will bind themselves with the active sites of the enzymes because more active sites will be available at that time. So due to which the reaction will start very fastly, but it will stop very early because there are many active sites due to which the reaction has been started very fast. So it will be stopped earlier. It will be finished because substrate concentration has been decreased at that time because all of the substrate has been changed into the product in a very short time. So reaction will be stopped. So here in the A, you can see the absorbance. The dotted line shows the reaction 2 and the other line show the reaction 1. So reaction 2 is, no, this is not, because it should be fast according to my explanation. So it is fast, but it is continued. No, it will not continue. It will not continue because if there is availability of more active sites, so the reaction will be stopped earlier. So B is also not a correct option. So C, you can see it is earlier, started earlier, and it stopped earlier as well. So C is the correct option. And in D, you can say it was slower than the first. No, it should not be. It should not be. I have explained that it will start fastly and stop earlier because there will be less concentration of substrate molecules as comparison to active sites available in the reaction. So answer is C. Now we are moving towards the next question, and next question is, protease are a group of enzymes that digest proteins. That digest protein. A bacterial protease that is secreted from a disease-causing bacterium could act as an antibody. So, which statement about protease are correct? First of all, the first statement is a bacterial protease that is secreted from a disease-causing bacterium could act as an antibody and cause the production of antigens against it. This is not a correct option. This is incorrect statement. That is, a bacterium protease secreted from a disease-causing bacterium would likely be an antigen itself. So antigens are substances that trigger an immune response leading to the production of antibodies. So this is not the correct statement. The second, non-competitive inhibition of a protease that has an optimum condition of pH 2 can be overcome by increasing the substrate concentration and increasing the pH. Statement 2 is also incorrect because the non-competitive inhibition of a protease cannot be overcome by simply increasing the substrate concentration. So non-competitive inhibitors bind to a site on the enzyme, um, anywhere on the, you can say, enzyme instead of active site. So reducing its activity, increasing substrate concentration won't replace the inhibitor. So it is also not a correct option. Water molecules are required when peptide bonds that link the monomers together are broken by the action of protease in hydrolysis action. Yes, this is the correct option because the water molecules are essential for the action of protease is because proteases catalyze hydrolysis reactions which involve the breaking of peptide bond using water molecules. So when water molecules are added into them, then at that time there will be the you can say breakdown or hydrolysis reactions occur. So only D is the correct option and third statement is the correct statement. Now the next question is, the diagram shows part of a cell surface membrane, and you can see this is the cell surface membrane, and question this is question number 16, and there is cell surface membrane. You can see the picture. 1. The which row correctly identify the molecules labeled 1 and 2? Labeled 1 and 2. And here you can see, students, they have given us glycoprotein, protein, it is protein, it is protein, and it is protein. But they have asked about the whole. So it is protein and it is carbohydrate. So this is the glycoprotein. This is glycoprotein. And this is the phosphate head. So answer is 1 is glycoprotein and this is the phosphate head. A, B, D are not the correct. Simple, very simple question. That is the glycoprotein. This is glyco and below is the protein. So the glycoprotein and it was glycolipid. This is glycolipid, but here a protein is beyond in the at the base, so it is glycoprotein. Next, uh, next question is the diagram shows the mean dimensions of an epithelial cell in micrometers. The width is 2 micrometer, the height is 3 micrometer, and length is 8 micrometer. So, which row shows the surface area to volume ratio of the epithelial cell and how the surface area to volume ratio of the cell would change if the cell width doubled. If cell width is doubled, the surface area will decrease. As the size increase, the surface area will decrease. So we have to find out the surface area decrease this way. So B and D are not correct option. A or C can be the correct option. So find the correct option. We have to calculate the surface area to volume ratio. So we have, first of all, we have to see the formula for volume. It is volume length into width into height. It is 8 into 2 into 3. It is 48 micrometer. Now we have to find out the this is volume. Now surface area. Formula for surface area is equal to W plus 2 length and height plus 2 width and height. So we will put the values. After putting the values, you can see that 8 into width, you can say that is 2 plus 2 into length is 8 into height is 3 plus 2 into width is 2 into height is 3. So after calculating, we will find 32 plus 48 plus 12. So it will be equal to 92 micrometer square. So surface area to volume ratio will be to volume ratio is equal to surface area is 92 and volume is 48. If we divide both of them with 4, so it will be, here you can see 12, and so answer is C. So answer is C. You have to use these formula to find out the answers, and answer C is the correct option. A, B, and D is not the correct option. So only C is the correct option. With the help of these formulas, you will be able to find out the correct answer. Now the next question is which diagram shows the correct direction of net water movement between the four cells due to osmosis. And you know, in osmosis, the water moves from its higher water potential towards the lower water potential. So they have shown this water potential sign. You can see A, B, C, and D. In A, you can see the water is moving from its higher water potential towards the lower water potential. So here you can see this one is at its higher water potential because water is moving towards other three. Four cells has been shown over here. So it means water is moving from this, and this. It means it is, and then second highest, this one from here is moving water, and lowest is this one. Now in the second picture, so it can be a correct option. Now move towards the B, and here you can see the water is moving from -16 towards the 32 and 36, and but it is also how a cell with low water potential can give the water, can move the water from the lower water potential towards the higher water potential. So this is not moving. B is not the correct option. C, in C you can see water is going in this direction or in this direction, and water is also going in this direction, and from this in this direction. Again, this is not the correct option. C, where the arrows are in correct direction. In D, you can see the water is also going in this one. Two arrows are shown, and one is, it means 1, 2, and then it is also moving is this time, but it is also moving in this way. So this is the wrong. Again, correctly see, students, that three arrows are moving from this side towards this side, and the cell is moving towards the end. It is also moving in this way and in this way. It means this cell is at higher water potential. So A is the correct option. Water will move always in the direction where it is in higher water potential. It will not move from lower water potential towards the higher water potential. So water will always move from its higher potential towards the lower potential. So A is the correct option. Try to understand, watch these direction of these arrows, then you will be able to understand it. An investigation was carried out into the effect of 4 different treatments on the permeability of the cell surface membranes and tonoplast of beetroot cells. Beetroot cell vacuoles contain a red pigment. This pigment cannot diffuse to the tonoplast or cell surface membranes. 1 cubic centimeter cubes were cut from beetroot tissues and washed in running water for 20 minutes to remove any pigment released from damaged cells. 2 cubes were then placed in each of the 4 test tubes containing different contents and observed for 5 minutes. Picture shows a correct explanation for the observation recorded for one of the treatments. Dilute hydrochloric acid. Contents of test tube stay clear. Membrane proteins have been denatured. Dilute hydrochloric acid will not denature it. So this is not the correct option. Ethanol contents of test tube turn red. Yes, turn red because the lipids, including membrane phospholipids, have dissolved. It can be a correct option because ethanol, you can say, it helps to dissolve the lipids. So it is used for the identification of lipids. So it can dissolve the lipids into it. So we can say that it can be a correct option. Water at 20 degrees C, contents of test tube stay clear. Membrane proteins have been denatured. 20 degrees centigrade will not denature. So water at 80 degrees centigrade, contents of test tube turn red. So 80 degrees centigrade will denature the enzyme, so the reaction will stop. So again, it is not a correct option. Only B is the correct option because ethanol is there. Ethanol is used to for the identification of the lipids. So this is the correct option. Now question number 20. And question number 20, you can see the photomicrograph show a stage of mitosis. Stage of mitosis. Here you can see this which stage has been shown over here. This is the cell and the chromosomes are present in the center of the cell. So it means this is the metaphase. This is the second stage too, which is metaphase. So what would be correct for the next stage in mitosis? Next stage, which will be anaphase. When the chromosomes are in the center, this is metaphase, but when the anaphase is started, the chromosomes uh, separate from each other. They are not attached with one another. They get separate from each other and they move towards the, because they have to move towards the opposite poles. So two sister chromatids remains attached. No, they will not attach. Any one of them can be the correct. So nuclear membrane reforming, no, it will not reform. It will not present in the anaphase. It will be formed in the telophase. So A is the correct option. So it was all about question number 1 to 20. Now wait for my second video and thanks for watching. But please like, share and subscribe my channel because in this way you will be able to get more papers for your Cambridge exam. Thank you once again.